Welcome to Lakshman Biology. Now we will see about dihybrid cross test cross. Dihybrid cross test cross. In last class we have seen about monohybrid cross test cross. Now we will see dihybrid cross test cross. If you observe the dihybrid cross, in dihybrid cross what we have did? We have crossed round yellow seeds with the green wrinkled seeds. Dihybrid cross is nothing but cross between individuals which are differ in two characters. See, there is a round seed dominant over wrinkled. Round seed is dominant over wrinkled. That is the first character. Next, second character, yellow color. Yellow color is dominant over the green color. We have crossed the round yellow color seed with the wrinkled green color seed. These are the genotypes. These are the genotypes in homozygous condition. Then what we have observed, we have seen all dominant plants in F1 generation which contain heterozygous dominant. Then if you are not see the dihybrid cross video, once again see the dihybrid cross video. After that we will see this dihybrid cross test cross. In F2 generation we can see the different variety of plants. Some are wrinkled green, some are round yellow, some are yellow wrinkled. Like that we can see different types of characters, different phenotypes you can observe in the F2 generation. In F2 generation there is a plants. F2 generation there is a some plants. These plants are contain the round yellow seeds. Round yellow seeds. Round yellow seeds. See, round yellow seeds. Round and yellow are dominant phenotypic characters. Hence, genotype may be homozygous dominant or heterozygous dominant. Round yellow seeds that we can get in the F2 generation, they may contain genotype RR, capital R, homozygous dominant. They may contain homozygous dominant or they may contain heterozygous dominant. They may contain homozygous dominant. They may contain heterozygous dominant. In dihybrid cross test cross, we can identify what is the result if they if they contain homozygous dominant, and what is the result we can identify simply in dihybrid cross. We can identify genotype of dominant plant. Then how to do the dihybrid cross? Here also same dihybrid cross. The F2 generation dominant plant. Here the F2 cross between the F2 generation dominant plant. Dominant plant means here round yellow color seed plant. Round yellow color seed plant. The F2 generation cross between the F2 generation dominant plant with the homozygous recessive plant. With the homozygous recessive plant. Homozygous recessive plant for both alleles. Homozygous recessive plant means here the wrinkled green. When you cross the homozygous dominant plant with the homozygous recessive plant, not homozygous, dominant plant simply. F2 generates dominant plant if you cross with the homozygous recessive plant. Homozygous, all plants which contain wrinkled green colored seeds are homozygous recessive only. That we know the genotype because which contain recessive character. When you cross, what happened, that we have to see first. In case if the dominant plant contain heterozygous condition, what happened? See the dominant plant means round yellow seeds. there is a two possibilities. There is a two possibility of genotypes in round yellow seeds. One is heterozygous dominant, like this, heterozygous dominant. Another one is there is a chances for homozygous dominant, homozygous dominant. In case if it is the heterozygous dominant, what is the result that we will get? That we will see first. This is the homozygous recessive plant that we have to cross. Here, homozygous recessive plant will form the one type of gametes. All gametes contain small r, small y. If you see the heterozygous dominant plant, where if it is the round yellow plant is heterozygous dominant, which will form the different types of gametes. These are the possible gametes ry. There is a chances for ry. These two, if it combine, capital R, capital Y. These two, if it is combined, capital R, small y. These two, if it is combined, small r, capital Y. Small r, small y. These are the four different gametes can be formed. Few gametes contain both dominant alleles. Few gametes contain one dominant allele, one recessive allele, like this. Then, what are the genotype of F1 generation plant? Capital R, capital Y, small y. That are one type of plants. Second possibility, capital R, small y, small y. That is the second possibility. Next, capital R, small r, capital Y, small y. That is one possibility. Small r, small y. That is one possibility. We can see the formation of four variety of plants. Few plants are contain round yellow color seeds. Few plants contain round yellow color seeds. Few plants contain round green seeds. Few plants contain round green seeds, which contain heterozygous dominant condition for the seed shape, homozygous recessive condition for the seed color. Then few plants contain wrinkled yellow. Few plants are wrinkled yellow, and a few plants contain the seeds. Those are wrinkled green. All those are formed in 1:2, 1:1:1:1 ratio. Four types. If the newly formed plants contain four different phenotypes in 1:1:1:1 ratio, the plant that we want to identify its genotype is heterozygous dominant. In case if the plant, this F2 generation dominant plant contain homozygous dominant condition, which will form only one type of gametes. All gametes are with capital R, capital Y. Then the homozygous recessive plant also will form only one type of gametes. All gametes are with small r, capital Y, small y. Then we can see the formation of genotype which contain all heterozygous plant. Means if after the crossing of F2 generation dominant plant with the homozygous recessive plant, if you get all dominant plants, that will indicate all dominant plants means all seeds are round and yellow, which will indicate the given dominant plant for the test cross is homozygous dominant genotype. See, if all after crossing, if all dominant plants, that will indicate the test crossed plant is homozygous dominant. In case if result is 1:1:1:1 ratio, the plant which we are going to identify the genotype is heterozygous dominant. That is about test cross for the dihybrid cross. Sometime there is another chances also you can observe that chances if the given plant is, see, if you want to identify the genotype of the plant which contain yellow seeds, plant which contain yellow wrinkled seeds. Sometime there is a heterozygous condition for only one genotype. If the plant contain yellow wrinkled seed, there is a yellow color may be because of both capital Y's, or yellow color may be because of one capital Y, one small y. If you do test cross for this genotype, we will get the result. The result, what we will get? The result is this is the cross. We will get the result 1:1 ratio. Two types of plants are formed that are in 1:1 ratio. Then if the genotype of the F2 generation plant is heterozygous dominant condition for the seed shape, then if you do test cross for this, we will get the result. It is also in 1:1 ratio. Next, if the plant is, already we have seen. Okay, that is about the dihybrid class test class. Thank you.